Introduction to Molecular Biology. Exploring the fundamental. What is molecular biology? Study of biological processes. Molecular biology explores the inner workings of biological systems, from the molecular interactions within cells to the complex processes that govern living organisms. Molecular level analysis. It involves analyzing and understanding the molecules that make up living organisms, such as DNA, RNA, proteins, and other biomolecules. Unraveling life's mysteries. Molecular biology helps us unravel the secrets of life by studying how molecules work together to drive cellular functions, development, genetics, and evolution. Interdisciplinary field. It draws upon knowledge from various disciplines, including genetics, biochemistry, biophysics, cell biology, and computational biology, to gain a comprehensive understanding of molecular processes. Applications in medicine and biotechnology. Molecular biology plays a vital role in medical research, disease diagnosis, drug development, genetic engineering, and biotechnology advancements. Key concept of molecular biology. DNA, deoxyribonucleic acid. DNA is the hereditary material that contains the genetic instructions for the development and functioning of all living organisms. It consists of a double helix structure made up of nucleotides. Each nucleotide is composed of a sugar molecule, deoxyribose, a phosphate group, and one of four nitrogenous bases, adenine, thymine, cytosine, or guanine. Genes. Genes are specific segments of DNA that code for the synthesis of proteins, the building blocks of life. They carry the information needed for the inheritance of traits from one generation to the next. Each gene has a unique sequence of nucleotides that determines the sequence of amino acids in the protein it encodes. RNA, ribonucleic acid. RNA is a single-stranded nucleic acid molecule that plays a crucial role in protein synthesis. It acts as an intermediary between DNA and proteins and helps in the translation of genetic information. There are different types of RNA, including messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA, each with specific functions in the cell. Proteins. Proteins are complex macromolecules that perform a wide range of functions in cells. They are composed of amino acids and are involved in structural support, enzymatic reactions, cell signaling, and more. The sequence of amino acids in a protein is determined by the sequence of nucleotides in the corresponding gene. DNA. James Watson and Francis Crick discovered the deoxyribonucleic acid, DNA, structure in the cell in 1953 using X-ray diffraction patterns, which showed that the DNA molecule is long, thin, and DNA is the hereditary material that contains the genetic instructions for the development and functioning of all living organisms. It is found in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. DNA has a double helix structure composed of two strands twisted around each other. Each strand is made up of nucleotides, which consist of a sugar molecule, deoxyribose, a phosphate group, and a nitrogenous base. The four nitrogenous bases in DNA are adenine, A, thymine, T, cytosine, C, and guanine, G. The bases on opposite strands are paired together, A with T and C with G, forming complementary base pairs as a spiral-like shape. DNA. The complement of a DNA sequence consists of complements of its bases. The DNA therefore consists of two complementary strands which bind to each other tightly, providing a stable structure. This structure also provides the means to replicate, in which the double DNA helix structure is separated into two strands, and each of these strands are then used as templates to synthesize their complements. The DNA molecule is wrapped around proteins called histones into complex wall structures called chromosomes in the nucleus of each cell. The number of chromosomes depends on the type of eukaryote species. Each chromosome consists of two chromatids, which are call-shaped structures connected near the middle forming an X-like structure. Chromosomes are kept in the nucleus of a cell in a highly packed and hierarchically organized form. A single set of chromosomes in an organism is called haploid, two sets of chromosomes is called diploid, and more than two sets is called polyploid. Humans are diploid where each chromosome is inherited from a parent to have two chromosomes for each of the 23 chromosome set. The sex chromosome is chromosome number 23, which either has two chromosomes shaped X resulting in a female, or has X and Y resulting in a male. The type of chromosome inherited from father determines the sex of the child, in this case, genes. A gene is the basic unit of heredity in a living organism determining its character as a whole. A gene physically is a sequence of DNA that codes for an RNA, mRNA, tRNA, or rRNA, and the mRNA codes for a protein. The study of genes is called genetics. Gregor Mendel in the 1860s was first to experiment and set principles of passing hereditary information to offspring. There are 23 pairs of chromosomes in humans, and between 20,000 to 25,000 genes are located in these chromosomes. The starting and stopping locations of a gene are identified by specific sequences. The protein coding parts of a gene are called exons, and the regions between exons with no specific function are called introns. Genes have varying lengths, and also, exons and introns within a gene have varying lengths. A gene can combine with other genes or can be nested within another gene to yield some functionality, and can be mutated, which may change its functionality at varying degrees, in some cases leading to diseases. The complete set of genes of an organism is called its genotype. Each gene has a specific function in the physiology and morphology of an organism. The physical manifestation or expression of the genotype is the phenotype, which is the physiology and morphology of an organism site. A gene may have different varieties called alleles resulting in different phenotyping characteristics. Humans are diploid, meaning we inherit a chromosome from each parent, therefore we have two alleles of each gene. The genes that code for proteins constitute about 1.5% of total DNA, and the rest contains RNA encoding genes and sequences that are not known to have any function. This part of DNA is called junk DNA. There is no relatedness between the size of genome, number of genes, and organism complexity. In fact, some single-cell organisms have a larger genome than humans. RNA. The ribonucleic acid, RNA, is an important molecule that is used to transfer genetic information. It has a similar structure to DNA but consists of only one strand and does not form a helix structure like DNA. It also has nucleotides which consist of a sugar, phosphate, and a base. The sugar, however, is a ribose instead of deoxyribose, and hence the name RNA. Also, DNA base thymine, T, is replaced with uracil, U, in RNA. The fundamental kinds of RNA are the messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA, which perform different functions in the cell. RNA provides a flow of information in the cell. First, DNA is copied to mRNA in the nucleus, and the mRNA is then translated to protein in the cytoplasm. During translation, tRNA and rRNA have important functions. The tRNA is responsible for forming the amino acids which make up the protein, as prescribed in the mRNA, and the rRNA molecules are the fundamental building blocks of the ribosomes, which carry out translation of mRNA to protein. Types of RNA. Messenger RNA, mRNA, Messenger RNA carries the genetic information from DNA to the ribosomes for protein synthesis. It is transcribed from a DNA template during the process of transcription. mRNA serves as a template for translation, where the information in its nucleotide sequence is converted into a specific sequence of amino acids to form a protein. Transfer RNA, tRNA. Transfer RNA is responsible for transferring amino acids to the ribosomes during protein synthesis. Each tRNA molecule has an anticodon region that is complementary to a specific codon on the mRNA. The anticodon ensures that the correct amino acid is added to the growing protein chain. Ribosomal RNA, rRNA. Ribosomal RNA is a major component of ribosomes, the cellular structures where protein synthesis occurs. rRNA helps in the catalysis of peptide bond formation and provides structural support to ribosomes. It also assists in positioning the mRNA and tRNA during translation. Small nuclear RNA, snRNA. Small nuclear RNA is involved in the processing of pre-mRNA molecules, which are transcribed from DNA. It plays a role in the splicing of introns, non-coding regions, and the joining of exons, coding regions, to form mature mRNA. MicroRNA, miRNA. MicroRNAs are small RNA molecules that regulate gene expression by binding to complementary sequences on mRNA. They can inhibit translation or promote mRNA degradation, thus controlling the amount of protein produced from specific genes. Long non-coding RNA, lncRNA. Long non-coding RNA refers to RNA molecules that are transcribed from DNA but do not code for proteins. They have diverse functions, including regulation of gene expression, chromatin remodeling, and organization of nuclear structures. RNA function. mRNA carries the genetic information from DNA to the ribosomes, where it is translated into a specific protein. tRNA acts as an adapter molecule, carrying specific amino acids to the ribosomes during protein synthesis. rRNA forms the catalytic and structural core of ribosomes, facilitating the assembly of amino acids into proteins. Importance of RNA. RNA is involved in regulating gene expression, controlling cellular processes, and playing a role in various diseases. RNA-based technologies, such as RNA interference, RNAi, are being utilized for gene, protein. Proteins are large molecules of the cell, and they carry out many important functions. For example, they form the antibodies which bind to foreign particles such as viruses and bacteria. As enzymes, they work as catalysts for various chemical reactions. The messenger proteins transmit signals to coordinate biological processes between different cells, tissues, and organs. Also, they transport small molecules within the cell and the body. Proteins are made from the information contained in genes. A protein consists of a chain of amino acids connected by peptide bonds. Since such a bond releases a water molecule, what we have inside a protein is a chain of amino acid residues. Proteins have highly complex structures and can be analyzed at 4 hierarchical structures. The primary structure of a protein is specified by a sequence of amino acids that are linked in a chain, and the secondary structure is formed by linear regions of amino acids. A protein domain is a segment of amino acid sequences in a protein which has independent functions than the rest of the protein. The protein also has a 3D structure called tertiary structure, which affects its functionality, and several protein molecules are arranged in quaternary structure. The function of a protein is determined by its 4-layer structure. A protein has the ability to fold in 3D, and its shape formed as such affects its function. Using its 3D shape, it can bind to certain molecules and interact. For example, mad cow disease is believed to be caused by the wrong folding of a protein. Technique of molecular biology. Polymerase chain reaction, PCR. PCR is a widely used technique in molecular biology to amplify a specific DNA sequence that allows for the production of millions of copies of a target DNA segment. PCR involves a series of temperature cycles that facilitate DNA denaturation, annealing of primers, and DNA synthesis by a heat-stable DNA polymerase enzyme. PCR has revolutionized various fields, including genetics, forensics, medical diagnostics, and research. DNA sequencing. DNA sequencing is a technique used to determine the precise order of nucleotides in a DNA molecule. It provides valuable information about gene structure, genetic variations, and evolutionary relationships. Various methods of DNA sequencing have been developed, including the Sanger sequencing method and more recent next-generation sequencing (NGS) technologies. DNA sequencing has had a profound impact on genomics research, personalized medicine, and understanding genetic diseases. Gel electrophoresis. Gel electrophoresis is a technique used to separate and analyze DNA, RNA, or proteins based on their size and charge. It involves placing the molecules in a gel matrix and applying an electric field to move them through the gel. The molecules migrate through the gel at different speeds, resulting in separation based on size or charge. Gel electrophoresis is a fundamental tool in molecular biology for DNA fingerprinting, genotyping, and analyzing gene expression. Recombinant DNA technology. Recombinant DNA technology involves the manipulation and transfer of genetic material between organisms. It allows for the creation of genetically modified organisms, GMOs, by introducing specific genes into an organism's genome. Techniques such as DNA cloning, restriction enzymes, and DNA ligases are used in recombinant DNA technology. This technology has revolutionized fields like agriculture, medicine, and biotechnology. Gene expression analysis. Gene expression analysis techniques allow researchers to study how genes are activated and produce proteins. Methods such as reverse transcription, polymerase chain reaction, RT-PCR, microarrays, and RNA sequencing are used to measure gene expression levels. These techniques help in understanding cellular processes, development, disease mechanisms, and identifying potential drug targets. Application of molecular biology. Genetic engineering. Molecular biology techniques enable the manipulation and transfer of genetic material between organisms. This has paved the way for the development of genetically modified organisms, GMOs, and advancements in biotechnology. Genetic engineering has various applications, such as creating crops with enhanced traits, e.g., insect resistance, drought tolerance, and producing pharmaceuticals through recombinant DNA technology. Disease research and diagnosis. Molecular biology plays a crucial role in understanding the genetic basis of diseases. It helps in the diagnosis, prognosis, and development of treatments for genetic disorders, cancers, and infectious diseases. Techniques like DNA sequencing and polymerase chain reaction, PCR, aid in identifying disease-causing mutations, monitoring disease progression, and assessing the effectiveness of therapeutic interventions. Forensic analysis. Molecular biology techniques are extensively used in forensic science for identifying and analyzing biological evidence. DNA profiling, based on analyzing specific regions of the DNA, is a powerful tool for criminal investigations, paternity testing, and establishing human identity. The accuracy and sensitivity of molecular biology methods have greatly improved the reliability of forensic analysis. Evolutionary studies. Molecular biology provides insights into the evolutionary relationships between organisms by comparing DNA and protein sequences, Scientists can trace the evolutionary history of species and understand their genetic divergence and relatedness. Molecular phylogenetics helps in reconstructing evolutionary trees and unraveling the patterns and processes of evolution. Drug development. Molecular biology techniques contribute to the discovery and development of new drugs. Understanding the molecular mechanisms underlying diseases helps in identifying potential drug targets and designing therapeutic interventions. Molecular biology tools are utilized in drug screening, target validation, and studying the pharmacokinetics and pharmacodynamics of drugs. Be curious.